Okay, so the first thing we want to do is to go into the terminal, which will load up the metagenomics topic GitHub repo, which has the data that we're going to use as practice here. So the goals of this demo are to demonstrate the usage of Kraken 2 for taxonomic classification using k-mers. Second, we're going to demonstrate the usage of a program called BB-Duke, which is in the BB-Tools package to do adapter trimming using k-mers. And the third is to demonstrate the usage of Krona for visualization of these classification results. So due to time, space, and memory constraints on Binder, we're going to use the Silva 16S RNA database rather than a more complete database, which would contain entire genomes. And what that's going to mean is that we're not going to have very many hits because we're going to be using raw metagenomic reads. So the first thing we're going to have to do, is install Kraken 2. The version that's on Conda isn't working. It's a little outdated, and it uses FTP, and Binder doesn't allow you to install things with FTP, so we have to install it from scratch. Okay, so what Kraken is doing, it just installed itself. It downloaded these Silva taxonomic files and is now building the database. Okay, there we go. Now we just have to add a couple of things to our path here, which tells our bash where we can find the Kraken programs and also where we can find the Kraken database that we just built. So if we look in that, we can see it built a database called Silva, which has these various files that are needed for the database. If you're building the standard database, I think it said it took a couple hours and did about 100 gigabytes of raw data to build that database. So now we're going to go into our data directory where we have some raw metagenomic reads here, and just try running Kraken on a couple of these reads. So Kraken 2, if we run it without anything, it'll show us the help. So the most important things that we need to run Kraken are we need to tell it what database to use. You can build as many databases as you want. They each have a unique name. So in this case, our database is going to be called Silva. And then if you have a bunch of threads, you can tell the Kraken 2 how many threads you want to run it with. There's a quick mode of operation, which just uses the first hit or hits to make a determination. Otherwise, Kraken will use all of the k-mers that match to figure out what the identification of each sequence is. You can also do things like have it put classified and unclassified reads into different files so that you can separate those out. We'll also need to tell it if we want to output the file into a specific place, the raw output. We can also give it a confidence value in the range of 0 to 1, where the confidence is based on the number of identified reads over the number of total reads for each taxonomic group. We'll go through a couple of these. There's different output formats, a basic report and one that matches up with Metaflan. You can use paired reads or single reads. And then you can use various compressed groups or not. And so we're going to start by running it just with one sample on the most basic setting. So we're going to tell it which database to use, and then we're going to pick one of these and just run it. So this is the raw output. It just puts it to the standard out, and unless you want it to go somewhere else, in which case you can use the double dash output flag. And what we see is that the results here, it ran this sample. It identified 86,405 reads in this sample that it processed in 5.5 seconds. Of those, it classified 85.52% of them, and it did not classify 12,500 of them. I'm just going to go through the data report here real quick. So if you just take a look at each line of this, there's a few different columns here. The first one, in this case, shows C, that means classified. Here's one U that's unclassified. If we look at one of the classified ones, the next column shows the name of the read. So if you were to look at the FASTQ file that you put into this, it would have this read name show up. The third column is the NCBI taxonomic ID, or whatever taxonomic ID was in the database you used, in this case, the Silva one, of the read that it so classified. Then in the next column, it shows the read length. This is 301 base pairs. These are MySeq reads. And then it shows you an overview of which k-mers classified to which taxonomic ID. So in this case, the first 93 k-mers, and in this case, it's using a default k-mer length of 35 base pairs, classified to a tax ID of 0, which is the root of the system. The next 5 classified to tax ID 1. The next 40 again to 0, and then there was 1 that classified to taxonomic ID 44130, and then another 128 that classified to the root. So based on this, Kraken decided that the taxonomic classification was to this, this tax ID 44130. And you can see that a lot of these reads classified to this, this taxa 44130. So let's do it again, but with a somewhat nicer report format. And then we'll talk about what this means. So I'm going to use -- report and then call it Kraken2 report. 3, and then use the same. And this time I'm going to redirect the output, the raw output, this stuff up here, just to dev null so that I don't have to look at it. Check the text. Okay, so it did the same thing. It ran exactly as it did before, but in this time it output a report that we can look at here. So on the left, we have the percentage of sequences that were classified to each taxonomic level. The next column is the number of reads that were classified to each level. The third column is the number of unique reads, I think. Actually, I have to look that up. The fourth column is the taxonomic level. So this is unclassified, R is root, D is domain, P is phylum, class, order, family, genus, ... And then the next column is the taxonomic ID again, and then the name of the group indented to look at the to match the taxonomic level here. So if we scroll through this, we can see that there's a few IDs to various things here: bacteria, diatoms, eukaryotes. But the vast majority of them here, 80% of them, classified to this group in the Microcalaceae, within the CFB bacteria. And this is a little fishy because these are metagenomes, so we would really not expect things, so many reads to classify to a database that only contains 16S ribosomal RNA genes. So why might that be? Well, to take a look at that, let's try cleaning up the reads a little bit. And to do that, we'll first just do the very minimum, which is to trim the adapters off. And I meant to make a little picture of this, but when you do Illumina sequencing, there's several stages that involve adding primers, adapters, and indexes to the original reads that you amplified or that you extracted and then sheared. And if you have a small shear length, for example, if your sequences that go into the library prep are shorter than the read length of the instrument you're using, then what you can have is read through, where on the right-hand side of the read, it can actually read into the primer and the adapter and everything on the opposite side. And so that's bad. It's a mess, and it makes it bad for downstream analyses. So one thing that we always want to do is to trim off those extra adapters and everything if they exist. So a program that we're going to use to do that in this case is called BVDuke, which is written by Brian Bushnell at JGI, and is part of a whole suite of tools in the BB Tools package. It's watching things for me. A couple of more that we'll probably use in the Metagenomics curriculum are BBMap for mapping reads, BBmerge for merging reads, BBnorm for normalizing reads. We'll do some sketching using BB Sketch and SendSketch. So there's a lot of programs. I think there's like 150 or 200 programs that are in this package dealing with basically anything you can think of to do with manipulating DNA sequences. So as I mentioned, we're going to use BBDuke. BB Tools has a particular format for all the flags, and they are comprehensive, so there's usually dozens of flags that you can choose for any particular program, although generally you won't need most of them. There's only a few that you'll need. So if we run this using just the very basic commands, what we need are some input files, some output files, and then a reference file that contains the contaminants we want to remove. In this case, BB Tools comes with a built-in FASTA file containing all known Illumina adapters, and probably for other technologies too, that we can use as the default. If you're using some custom or proprietary indexing or kit or something, then you might have to add it yourself if you want to remove those sequences. So let's try just, again, this one sample that we were working with before. We'll rename it to a trimmed version. And then we'll tell it which sequences to use. You can also put in literal sequences here. If you have a single barcode you want to remove or something, you can just type it there and it'll be removed. And then we want to tell it to trim from the right. So it'll start trimming those adapters that have been read through. And the BB tools are usually very fast, and they're written in Java. It's really nice that you don't have to install anything. They just work. So I end up using them for a lot of things rather than trying to go through the rigmarole of installing some new software. So here's the output from the BB Duke. We input 86,405 reads again, 26 million base pairs. Of those, it trimmed most of them, 94%, and it left us with 40% of the bases. So in this case, it trimmed off 60% of all of the DNA in there, which means in this case that these libraries were not that great and that the insert size was smaller than ideal. So we're losing a lot of data here because of that. Okay, so now that we have some cleaned up reads, which we can just take a look at here. So we see our reads are there, but now they're different lengths because the right-hand side was trimmed based on the Kamer trimming. So if we run our Kraken command again, using these trimmed reads, we'll see what's different. So before, 90-some% of the reads were classified, and this time only 246 were, 0.29%, which seems to me a much more reasonable number. So if we look at that file, we see that all of those CFB are gone, and that's probably because it's because in the database, the Silva database that they used here, that sequence for one of those organisms must have an adapter in it. So someone might want to tell them that they have adapters in their sequences so that they can trim those and remove them from the database. So now if we look, we can start to see what might actually be in these samples. Microbia, some fungi, some diatoms, etc. So this looks more like what we'd expect. There's only a few reads matched because the 16S ribosomal RNA gene is only a small fraction of the entire genome. And this also is pretty obviously a seawater sample. We have SAR11 here. So everything looks pretty good there. Okay, so the next thing that we can do is to try out the confidence flag and see how that affects it. So we can just go to our same command here, add the flag confidence, and pick a number like 0.5. It has to be between 0 and 1. Run it again. And see, this time less than half of the number were classified as before. So in this case, only 102 out of the 86,000. So you probably want to look at your own data sets and find the confidence value that's right for you. I'm actually not sure what they're recommending in this case, but higher is probably better if you're trying to be very specific. For example, if you're actually doing 16S analyses with these, whereas if you're doing metagenomic classification, lower might be okay if you're just trying to get a genus or family level identification for whatever you're working with. There's also another report style, the MPA style, which we can run real quick. And that puts each taxa on an individual line with the number. So there's only 2 columns here. It can be useful for your downstream pipelines if they already use Metaflan, for example. Okay, I might take a pause here and ask if anyone has any questions before I go on about what we've done so far. You just unmute yourself if so. Okay, if not, I'll keep going. The next thing I wanted to talk about was some more options within BBDuke. So we used the very basic options for the first trimming, but there's a lot of other options that we can go into. So the options that, that the author of the tool recommends as a default for trimming Illumina adapters is, like we said before, to use the built-in adapters file, ref equals adapters, to trim from the right, to use a k-mer length of 23. So in this case, it builds the index using 23 mers and then compares your sequence against that. But it allows a smaller k-mer of 11 base pairs at the end of the sequence, where it will also match. You can also ask it to look for a mismatch. So hdist in this case is Hamming distance, a 1 base pair difference between the expected k-mer and the observed k-mer. This slows things down a little bit. You can increase this, but as you do, you're more likely to trim things that aren't actually the target. There's another command here, TBO, which means trim by overlap. In that case, if you have paired end reads, it will overlap them and find where the actual target sequence is and where the adapters are by looking at where those sequences overlap, and then trim off the edges where they don't match. A related command is TPE, trim both pairs, trim paired ends, which means to trim both to be the same length. In the case that there is an overlap and you miss a part of the adapter for some reason, that'll make sure it's gone. And then BB Tools is conservative. It doesn't like overwriting files unless you tell it to explicitly. So OW means overwrite, and this says allow overwriting of existing files. It's for future references. There's also a number of commands that are involved in quality trimming. So in this case, we're only looking at the actual sequence and not at the quality sequences that are also in the FASTQ files. And so we'll talk more about that in another lesson where we talk about quality trimming. You saw a little bit of it with Alexis's tutorial on FastQC and multi-QC. And so there are ways to look at that data and choose some nice parameters for cutting off your DNA sequences based on the quality scores of each sequence. But we're not going to go into that at this point. So I want to redo the BBDuke trimming, and what we want to do is trim all of our samples so that we can run them all through Kraken. We'll start by just demonstrating the use of the paired end side of things. So in this case, we'll have 2 inputs. We'll have the forward reads, the read 1 here, and then input 2 is the reverse reads right there. So then we're going to have also 2 output files. We're going to have trimmed read 1 and trimmed read 2. And then we're going to use all of the recommended flags. So using the adapters file again, trimming from the right, k equals 23, min equals 11, h_dis equals 1, trim by overlap, and trim paired ends. And then we'll go ahead and run that. We got our results. It's about the same as before, about 40% returned. And here we have our trimmed files. So we can run these once more through Kraken using the paired end flag here. We'll give it both the forward and the reverse trimmed reads, and then take a look at the report. So again, the vast majority of sequences were unclassified, as we would expect based on the database we're using, and we see the same things show up. So it works. There are probably some nice packages for looking at the Kraken output, but if you want to do some command line filtering, we can do some of that. So I'll just show you a couple of things that you might want to take a look at here. One is to sort the data based on the relative abundance. So say we want to just look at the most abundant things, we can sort by the number of reads that were classified, which is in the second column. And so in Bash, we'll use the sort command. K means the key that we're going to use, and so that's column 2 and only column 2, that's the comma 2. We're going to sort in reverse, that's R, so it'll use higher numbers first, and N means numeric, so we're going to look at the numbers and not the ASCII character code to sort them for. So if we wanted to see the most abundant things we were classified here, we run that command, and we see that, again, the top one is unclassified, root, 190 were classified to bacteria, 165 to eukaryotes, and then we can see the different taxa here and how many were classified. There they ask, is there a way to extract out the positive Kraken hits? Yes, in Kraken 2, there's a command classified out, where you can actually print the classified sequences to a file, and we can test that out real quick. Probably need to put this before. Okay, I haven't tested this, so we're going rogue here. It might not work with the paired ends. So here are our classified reads now. Great. Okay, so the next thing we want to do is to put all this together and do the trimming for each set of paired end reads, and then run them through Kraken to produce our report. So we got into loops a little bit, for loops with Lexis. So we're going to use a for loop again. In this case, because we have paired ends with common names, excuse me, we can use this to pick out individual ones. So this is the way I usually do it. I list all of the files that I'm going to be working with. And then I use the cut command to trim off, in this case, the first field based on the underscore character, which means it will return this part right here. And then I sort those and then use this -u flag in sort, which only gives me the unique ones. So in this case, it produces the first part of each of these different sequences, and then this trimmed one, because it has an underscore there too. So we're just gonna remove those for now, because they will be rebuilt as we do this. And then we're gonna use this for loop to go through each one. And just to make sure it works, we can try this out for, we'll call the variable prefix in this command, the return value from this command, do something. In this case, we'll just echo it, and then we'll call it done. So as it goes through the loop, it takes the value for each one of these, it puts it into echo, and then it returns it to the screen and finishes. So now we'll just add the next step here, which is to get the name of the forward and the reverse read from each of these. And the way we'll do that is to use this arrays in Bash, which are produced when you use the parentheses here in the variable assignment. And we're going to do that to get this globbed value for each one of them. So we're using the asterisk here as a glob to match whatever's in the middle between this prefix that we found and the end of the string, and use that downstream. So if we try this first one, read one, and then look at it, it will hopefully be the full file name, and then same for read two. Very good, we have our file names here. And then the first thing we'll do in this loop is bbduke. So again, we'll put in read one, forward and reverse. We'll put in some output files, which will just prepend the word trimmed to each of those. And then we'll do the same thing here with the references and the trimming. Then we'll run Kraken 2 on the trimmed files using the Silva database. We'll output a report called Kraken 2 report paired, and then the prefix here to make each file unique. And then again, we'll put in the trimmed file names here and there. And then that'll be done. So I'm just going to copy and paste this into. and it'll take a couple minutes here probably. Okay, so we finished all of our sampling. We got all of our reports and all of our samples trimmed. And so we could keep looking at these on the command line, or we could get a little fancy. And so we're going to get a little fancy and install Krona. So Krona is a very popular package for looking at this kind of data because it's explorable. You can kind of drill into your data and look at it in a pretty simple way. So Krona was already installed using Conda. So you can see it comes with a number of different commands for importing your data from basically whatever source you have into its own format. And so the command we're going to use first is kt update taxonomy, which is going to use the Silva taxonomy that we installed for Kraken 2 so that all of the taxonomic IDs that are in these report files will be recognizable by Krona. So we're going to run it using this flag only build, and then we're going to tell it where to find the taxonomy files, which Kraken downloaded. It runs very quickly because it's a small file. And then we can tell Krona in the next step where to find that taxonomy file so that it can use it in the next step. So the command we're going to use is kt import taxonomy. And if you run it by itself, you'll see that this also has a number of options. We're going to give it the taxonomy file it needs, and then you also have to give it a couple more things. In this case, it's going to be pulling from this tab delimited file, so you need to tell it where to find the column of input to use as the taxonomy ID. And then either this -s, which tells it the score, or the -m, which tells it the magnitude. And we'll use the magnitude flag here because we'll use the absolute abundance of reads here. So our command, and then the other thing we have to give it is an output file name. So we'll do kt import taxonomy. And then, let's just run it on one example file here. And it'll be an HTML file that will be produced. So that we can open in a browser. We'll tell it where to find our taxonomy file. Oops. Using the -tax flag. And then we'll tell it which file we want to run it on. These are called Kraken 2 reports. Okay, so we run it on that. It outputs our test.html file right here, which we can go to on the browser on the left. If we double click that, it'll open up, but it won't work right away. We have to click this Trust HTML button, and then we get our chronograph. As you can see, most of it here is red, which means unassigned, which is natural, as we would expect. But we can zoom in and look at the rest of the classifications here. As we see before, there's things that you would expect probably from the ocean, like SAR 11, but then we also have this other stuff over here, which probably shouldn't be there. I don't know what they are. And we can explore this and dig down as we want. If you've ever used a chronograph, you know how this works, but it's pretty straightforward. Okay, so finally, we will run a loop again to go through each of our Kraken 2 report files. and make a chronograph for each one. Da 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 da. There you are. And we can take a look at it. All right, I think that's all for this lesson.